Transport through cell membrane. Membrane transport means the transportation of molecules through cell membrane. Basically, there are two types of transport mechanism. First, passive transport mechanism. Second, active transport mechanism. First, passive transport. Passive transport is the movement of molecules across the cell membrane from a region of higher concentration to a region of lower concentration. Passive transport does not need energy. It is also called diffusion. Diffusion, it is of two types. First, simple diffusion. Second, facilitated diffusion. First, simple diffusion. It occurs by two ways. First, simple diffusion through lipid layer. Second, simple diffusion through protein layer. First, simple diffusion through lipid layer. By this diffusion, only lipid-soluble substances like oxygen, carbon dioxide and alcohol can diffuse through the lipid layer. Second, simple diffusion through protein layer. In this type of diffusion, channel proteins are responsible for the transport of water and water-soluble substances like electrolytes to pass through the cell membrane. Second one, facilitated or carrier-mediated diffusion. Water-soluble substances having larger molecules cannot diffuse through the protein channels. Such substances pass through the cell membrane with the help of some carrier proteins. Therefore, this type of diffusion is called facilitated or carrier-mediated diffusion. By this process, the substances are transported across the cell membrane faster than the transport by simple diffusion. Glucose and amino acids are transported by facilitated diffusion. Factors affecting rate of diffusion. First, permeability of the cell membrane. Rate of diffusion is directly proportional to the permeability of cell membrane. Second, temperature. Rate of diffusion is directly proportional to the body temperature. Third, solubility of the substances. Rate of diffusion is directly proportional to the solubility of the substances. Special types of passive transport. In addition to diffusion, there are some special type of passive transport. Bulk flow, filtration, osmosis. Bulk flow. It is defined as diffusion of large quantity of substances from a region of high pressure to the region of low pressure. Filtration. It is defined as the movement of water and solutes from an area of high hydrostatic pressure to an area of low hydrostatic pressure. Osmosis. It is defined as the movement of water or any other solvent from an area of lower concentration to an area of higher concentration of a solute. Active transport. Active transport is the movement of molecules across a cell membrane from a region of lower concentration to a region of higher concentration. Active transport requires energy. Active transport works with the help of the carrier proteins. Carrier proteins of active transport. They are of two types, uniport, symport. Uniport pump. It is a carrier protein that carries only one substance. Symport pump. It is a carrier protein that carries more than one substance. Substances transported by active transport. Ionic form substances: sodium, potassium, calcium, hydrogen, chloride, and iodide. Non-ionic form substances: glucose, amino acids, and urea. Types of active transport. First, primary active transport. Second, secondary active transport. Primary active transport. By this method, sodium, potassium, calcium, hydrogen, and chloride are transported across the cell membrane. Sodium-potassium pump. It is a primary active transport. Carrier proteins of sodium-potassium pump. It has got 6 sites. 3 receptor sites for sodium ions on the inner surface of the carrier protein, 2 receptor sites for potassium ions on the outer surface of the carrier protein, 1 site for the enzyme ATPase which is near the sites for sodium. Mechanism of action of sodium-potassium pump. 3 sodium ions inside the cell bind to the receptor sites of sodium ions located on the inner surface of the carrier protein. 2 potassium ions outside the cell bind to the receptor sites of potassium ion located on the outer surface of the carrier protein. The binding of sodium and potassium ions to the carrier protein immediately activates the enzyme ATPase. This ATPase causes breakdown of ATP into ADP with the release of one high energy phosphate. This energy causes some sort of conformational change in the carrier protein, so that outer surface of the carrier protein faces inner side of the cell and the inner surface of the carrier protein faces the outer side of the cell. Now dissociation and release of the ions takes place. So sodium ions are released outside the cell and the potassium ions are released inside the cell. Electrogenic activity of sodium-potassium pump. Sodium-potassium pump moves 3 sodium ions outside the cell and 2 potassium ions inside the cell. Thus, when the pump works once, there is a net loss of one positively charged ion from the cell. The continuous activity of the sodium-potassium pump causes reduction in the number of positively charged ions inside the cell, leading to the development of negative potential inside the cell. This is called the electrogenic activity of sodium-potassium pump. Second, secondary active transport. It is the transport of a substance along with sodium by means of a common carrier protein. There are 2 types. First, sodium co-transport. Second, sodium counter-transport. First, sodium co-transport. Along with sodium, another substance is also transported by the same carrier protein in same direction simultaneously. Substances carried by sodium co-transport are glucose, amino acids, chloride, iodine, iron, and urate. Second, sodium counter-transport. Along with sodium, another substance is also transported by the same carrier protein but in opposite directions simultaneously. Some of the counter transport are sodium calcium counter transport, sodium hydrogen counter transport, etc. Special types of active transport. In addition to primary and secondary active transport, there are some special type of active transport called vesicular transport. They are first endocytosis, second exocytosis, third transcytosis. First, endocytosis. Macromolecules that cannot pass through the cell membrane either by active or by passive transport, such substances are transported into the cell by a process called endocytosis. It is of three types. First, Pinocytosis. Second, Phagocytosis. Third, receptor mediated endocytosis. First, Pinocytosis. It is a process by which macromolecules like bacteria and antigens are taken inside the cell. It is otherwise called cell drinking. Second, phagocytosis. It is a process by which particles larger than the macromolecules like larger bacteria, larger antigens and other larger foreign bodies are taken inside the cell. It is otherwise called cell eating. Third, receptor mediated endocytosis. It is the transport of macromolecules into the cell with the help of a receptor protein. Receptor protein is clathrin, which is present in the pits on the surface of the cell membrane. This process play an important role in transport of hormones, lipids, antibodies, etc. Second, exocytosis. In this process, substances are expelled out from the cell without passing either through active or passive transport. This is the reverse of endocytosis. Third, transcytosis. It is a transport mechanism in which an extracellular macromolecules such as several enzymes, proteins and antibodies enters through one side of a cell, migrates across cytoplasm of the cell and exits through the other side.